The information in this module is accurate and complete to the best of our knowledge. All recommendations are made without guarantee on the part of the author or the sponsoring institutions. The author and the sponsoring institutions disclaim any liability in connection with the use of this information.
Welcome. In the last lecture, we went on a tour of the history of genomics, from discovery of DNA and genes to the advent of genome technology. In this lecture, Genome Projects, we will talk about the development of technology and methodology that was used in the reconstruction of the entire genomes, including that of the human genome map. A quick and important reminder of the definition of the genome. It is a complete set of genes or genetic material present in a cell of organism. At the end of this lecture, the students will identify what is chromosomal walking and how it was used in defining disease genes, describe the clone contig shotgun method, describe the whole genome shotgun method, state the main milestones of the Human Genome Project, describe the goals of a 1,000 Genomes project, identify what was the advantage of whole exome sequencing, identify what is the difference between de novo and reference assembly. The quest for the genome maps did not start with the era of genomics. Before the times of massively parallel and next-generation sequencing, in the pre-genome era, there was in fact a very substantial movement to map chromosomes of bacteria and even some eukaryotes, such as Drosophila, mice, and human. These early efforts were not in vain and has provided a valuable foundation for the modern genomics. Specifically, there were two general types of genome mapping called genetic mapping and physical mapping. Both types of genome mapping could guide scientists towards the location of a gene or a section of DNA on a chromosome. However, they relied on very different information. Genetic mapping looks at how genetic information is shuffled during the reproductive cell division, called meiosis. As you may know, during meiosis, some of the homologous chromosomes exchange arm in the process called crossing over. Since genes are located on chromosomes, the different alleles located in the same loci will exchange between the chromosomes as well. Alleles are different variants of the gene that can be found in the same chromosomal locations. An example of alleles in a fruit fly called Drosophila melanogaster are shown on the figure. Vermilion eyes, yellow body, white eyes, miniature wing, rudimentary wing, etc. The first genetic map of a chromosome was made by crossing fruit flies and recording crossing over between phenotypic characters like eye color, wing, and body shape in a fruit fly in 1913. It was proposed that the frequency of crossing over between two genes could help determine their location on a chromosome. Very soon, there were elaborate genetic maps that placed many loci relatively to each other based on genetic distance measured in centimorgans, or percent of progeny containing crossing over between them per generation. Human genetic maps were also constructed, except that instead of experimental breeding like in Drosophila, human geneticists used genetic distances calculated from genetic pedigrees. Physical mapping is based on measuring the physical distance directly by estimating the number of nucleotides or base pairs between them. The first physical maps were made with restriction enzymes. Restriction enzymes always cut DNA at specific sequence of DNA, or restriction sites. After the DNA is cut in pieces, the DNA fragments can be aligned to each other using the restriction profile pattern generated by electrophoresis. Potentially, a full restriction map of the chromosome can be built in this manner. They will show the sequence of locations containing a particular restriction site, for example, GAATTC, throughout the chromosome. Constructing the genetic and physical maps had one very important application in human genetics. In 1980s, mapping was done in order to identify the causing genes for many human diseases. Dr. Francis Collins was one of the most famous masters of this process. He and his team discovered the locations of 3 important disease genes responsible for cystic fibrosis, CFTR gene shown on chromosome 7, Duchenne muscular dystrophy, and Huntington disease. The technique he used at the time was called chromosome walking. With time, it has become the basis of the first large genome projects, and Dr. Francis Collins was becoming the leader of human genome projects led by the National Institutes of Health. So let's look at the specific example here. You can see that chromosome walking is a method of positional cloning used to find, isolate, and clone a particular gene in the gene library. We use restriction profiles or restriction fingerprints, if you prefer to call them that, to align DNA fragments from clones in order to measure a distance between a known marker and a disease gene. At each step of a chromosome walk, the entire library is searched for a fragment that shares the restriction fragment of the last clone added to the walk. On the picture, the first subclone is shown in red, the next one is in orange, and the last one is in yellow. The matching is done sequentially in the direction of the chromosome walk. In practice, both types of markers, genetic markers and restriction fingerprints, can be combined in order to line up clones until a location of gene is reached. In principle, the chromosome walk can be used to assemble small genomes as well. First, the DNA is isolated, shared, and cloned. Then the clones are sequenced, and then finally, the clones are assembled into a complete sequence using the overlapping parts between the clones. The problem with the larger genomes in the 1980s and 90s was that it was perceived to be too computationally extensive to find overlaps between the sequencing clones. One obvious solution was to employ a hierarchical approach. Start with larger segments, map them to the existing genetic and physical map, and then keep mapping smaller subclones inside the larger ones. On the figure, we start with a genome map, made with markers at 200 to 500 kilobases apart. From the genome library, we pick a clone that matches the mapped segment. Then, we take the plasmid and break it into smaller pieces by sonication, or a combination of restriction enzymes. These short segments are sequenced, and since there is a reduced number of them, they are easily assembled into a contig. Recall that a contig is assembled from overlapping reads. The resulting contig spans the entire length of the cloned segment. So, as a result, we know the sequence of the mapped segment on the chromosome. As more and more mapped clones get sequenced, the blank spots on the genome map get gradually filled up. The Human Genome Project was a landmark genome project that is already having a major impact on research across the life sciences, with potential for spurring numerous medical and commercial developments. Based on the insights gained from the smaller genomes, such as yeast and worm, the HGP started in 1990 and was designed in 3 phases. First, the HGP relied on the physical and genetic maps established earlier, which served as a platform for generating and analyzing the massive amounts of DNA sequence data of the next shotgun phase. Second, the hierarchical shotgun. HGP divided human chromosomes into DNA segments of an appropriate size, which were then further subdivided into smaller, overlapping DNA fragments that were sequenced. Third, the finishing phase. This involved filling in gaps and resolving DNA sequences in ambiguous areas not obtained during the shotgun phase. This was a long and elaborate process and was supposed to continue for decades, but it did not take into account the rapid growth in technology development and the increased speed of computer capacity that would improve the assembly. At some point, some scientists started to believe that the computer power was sufficient to skip the hierarchical process, sequence the entire genome fragments at random, and let computers to do the assembly. So, at the end of 1990s, a private-led alternative was developed by Celera Incorporated, led by Craig Venter, called the whole genome shotgun approach. The Celera approach took multiple copies of the genome and fragmented them into 1 to 2 kilobase fragments that were sequenced without concern for what chromosomes were belonged to. Each sequenced nucleotide fragment was called a read. These reads were used later by the computer algorithms to reconstruct the original sequence in the process called Genome Assembly. These short reads were assembled into longer contigs based on their overlap. So, by definition, a contig is a continuous sequence of overlapping reads. Eventually, the two human genomes were published simultaneously in the prestigious journals Science and Nature. One by hierarchical shotgun approach by Francis Collins and his government-supported effort, and the whole genome shotgun assembled genome by the private group led by Craig Venter. The HGP was a 13-year-long, publicly funded, $3 billion project initiated in 1990 and completed in 2003, with the objective of determining the DNA sequence of the entire human genome. HGP was met with skepticism by many people, but the overwhelming success ushered a new era in medicine and led to the significant advances in the types of technology used to sequence DNA. By now, each dollar put in the Human Genome Project resulted in several hundreds of dollars economic returns. It turned out to be one of the most successful scientific investments ever made. The Human Genome Project was very expensive, but since that time, the price of individual genomes has gone down for several reasons. First of all, the technology has steadily improved and the data output of the sequencers has improved by incredible speed. At the same time, computer power has improved as well. There has been an exponential increase in the number of draft genomes of different species, such as chimpanzee, panda, cheetah, and of course the Puerto Rican parrot, which has been the cheapest genome ever made. At the same time, once the genome is well done, the others do not have to go through the intensive de novo assembly process. Instead, the reads can be aligned directly to the existing sequence. This requires a lot less of the data, so funds can be directed to resequencing many individuals of the same species instead. Resequencing is a term that can be used to describe two distinctive processes. One is use of resequencing for improving the quality of the existing DNA assembly for an organism. For example, the Human Genome Project, which was completed in 2003, provided the first fully assembled sequence of the human genome. Since then, scientists have been working to produce a reference sequence for a higher quality and accuracy. As a result, the human reference sequence has been vastly improved since 2003, with scientists correcting errors, rearranging the order of individual contigs, and filling out the remaining gaps in the sequence. Another use of resequencing is when we sequence many genomes. We can then compare the new genome sequences with that of the reference. In these studies, the focus shifts to genome variation. For example, if there is a base pair change in the new genome that isn't present in the reference genome, it may give us a clue to genetic origin of a particular trait or a disease. The availability of a reference human genome since 2003 has allowed for projects such as 1000 Genome Project, which launched in 2008, and it was the first project to sequence the genomes of a large number of people, at least 1000, to provide a comprehensive resource of human genetic variation. Scientists plan to sequence the genomes of at least 1000 anonymous participants from different numbers of ethnic groups within the following years, using newly developed technologies, which were faster and less expensive. It has been estimated the project would cost more than $500 million if standard DNA sequencing technologies were used. Therefore, several new generations of technologies were applied, lowering the expected costs to between $50 and $30 million. Another way to obtain information for less money is to focus only on the exomes. Exome sequencing is a transitional technology that has an excellent chance of finding variants responsible for inherited conditions at significantly lower cost than the whole genome sequencing. Whole exome sequencing is targeted to protein coding genes only. The exome represents less than 2% of genetic code, but it contains 85% of all known disease-related variants. Exome sequencing can efficiently identify coding variants across a wide range of applications, including population genetics, genetic disease, and cancer studies. This story is by no means finished. We're currently witnessing a revolution in genomic technology. Since the completion of HGP, the rise of the new generation sequencing technologies allowed faster and cheaper genomes. In fact, the speed of the genome sequencing has overcome the Moore's law, and there is no end in sight. In the next lecture, we will look at the next generation sequencing technologies, or NGS. Here are some review questions. This concludes today's discussion of the Genome Projects. In the next lesson, we'll be studying the topic of next generation sequencing.